Hello, and welcome to this critical video. Recently, the YouTube channel Adam Ruins Everything published another one of their contentious videos, this time on animal research with a focus on mice. Unlike most, this one has touched on something that is not simply a gross misunderstanding, but a deliberate partisan propaganda piece, as demonstrated in the first 5 seconds, where the protagonist's offsider states the show is about proving things wrong.
Adam, I've seen your show. It's about proving things wrong, but...
You will find a link to the original video in the description box below. To understand why this video is so very aggravating, it is important to realize that there is and will continue to be a very large gap between basic, preclinical, and clinical sciences. This gap is unlikely to be bridged any time in the foreseeable future. An example being this quote from Leonidas in 2006. Among 101 articles published between 1979 and 1983 in 6 top basic science journals that clearly made promises for a major clinical application of their findings in therapeutic or preventative interventions, only 27 technologies were evaluated in a published randomized controlled trial by 2003. For genetics, Leonidas had this to say: despite some concordance, correlation was still modest. Keep the fact that such research is very hard to turn from basic science into preclinical science into clinical science throughout the entirety of this video. The host Adam begins with the very first claim, that science is flawed.
Yes, it is, but the way we do science is often flawed.
Yes, science is a process of building successive blocks of research. Not every block works, and not everything is a road to further success. Science often has dead ends and pitfalls. These negative results are often unpublished and therefore unknown, meaning people repeat the same failures. Animal models, particularly mice, they have brain structures closely mimicking those of humans, and since they brought in ethics requirements, researchers are rarely allowed to dig around in human brains for purposes of research. A mouse is a damn good substitute in their place. This is also true of other biological functions, genetics, and pathologies. Sidekick gets it wrong by saying this. Everyone knows.
What? Everyone knows testing on rodents is practically the same as doing actual human trials.
It is not what is actually known. Mice and other rodents are used because they are a proof of concept, providing mechanistic and principal evidence. The rodent animal models are used for that exact reason, providing proof of concept and providing evidence of a line of reasoning. Human clinical trials are distinct from animal experiments. This is why researchers use genetically modified organisms, generally mice. The genes put into the mice correct much of the discrepancy in function and expression between species, and that of the 80% failures that are mentioned,
Out of all clinical trials based on mouse models, the results don't apply to humans over 80% of the time.
yes, experiments fail. Yes, clinical trials fail as well. This is called research. The higher rate is due to the complexity of the organism and the poor understanding of pathways, events, and other factors. Through the failures, the correct pathway and events can be extrapolated or elucidated. The process of building failed research explains dead ends. This is why Google Scholar has the slogan, "Stand on the shoulders of giants." But an 80% failure rate is not that bad when you consider that of all clinical trials, 90% fail. Further to this particular issue, at this point they introduced the BALB mouse.
Ooh, I've always wanted to be a BALBCJ laboratory mouse.
But that is not the most common model, and this video should be referring to the C57BL6, which is the most common due to its robust nature, easy breeding, wide availability, and well understood genome. The final part that is of particular concern at this stage is the filing.
Well, time to file these.
And although intended as humor, if a researcher ever filed their paperwork like this, they would open themselves up to a destruction of institution property charge. The lab reports, paperwork, materials, and pretty much any notes they made during the entire period of research are often property of the lab or institution they are working in, and they remain their property even if that person departs. The document trail provides protection and evidence to the place it is done in. This is in case of litigation or criminal charges. It also means that if a researcher is no longer around, someone else can pick up where they left off. Next is a particular comment regarding size.
You know, people are over 3,000 times bigger.
And size is not an issue when you are looking at whether or not something works the way it is theorized to do. The mouse is proof of concept and proof of mechanism because it is equally complex. It is a whole organism. Unless Adam is advocating that we return to the days of Tuskegee and Mengele, mice are the first port of call in research. They are small, easily maintained, cheap, and so similar to humans as to be practical. Next, they bring up the point of failed trials.
And those failed trials can cost hundreds of millions of dollars apiece. And this drives up the cost of development, meaning we get fewer new drugs.
Prior to even getting to trial stages, drug leads must be discovered, and this is often through public funding. Taxol is an example of this. The lead is identified, refined, and purified. This is then tested and sold, licensed, or other to bigger pharmaceutical companies with the resources to follow up. Failed leads are often part of bigger projects, having no great impact to the cost of the overall project and little to none on the license fee. The fee being the only part this applies to, and not the development costs. The effect on development means no cost of product from failed experiments. In fact, there are other reasons why drugs become expensive. The reason drugs are expensive is not because of the failed experiments that have preceded it. This argument is a drastic oversimplification of an extensive and deep series of issues relating to the development of any product within pharmaceutical sciences. The first reason is product development. This is touching on the failures of drug trials, already stated, but also other factors. A drug may exist for disease A, and this is typified by the Nature article seen here. Once these new targets are identified, pharmaceutical companies can either license their still patented drugs for use, or, if it is an out-of-patent drug, the discovery of its new application can be patented, and this means it needs licensing to be used in this novel way. The license and patents are what make new applications of old drugs expensive. New drugs are expensive because it takes 10 or more years to take them to market, and then a company may only have 5 to 7 years in which to recoup any costs associated with the new product before generic brands can be produced and the market usurped. Next is the cost of bringing a product to market due to the bureaucratic red tape. Authorities like the FDA have brought in more and more requirements and barriers to drug development and introduction to the market. This is exemplified by the comparison with the golden age of drug development. This is the period between the 1920s and the 1970s. The number and range of drugs developed and introduced in that window almost outstretch what has been made in the 50 years since. Despite the fact that we have greater access and greater means to develop these things, it is the end of that period that is marked by the thalidomide scandal that explains why these new rules are brought in, but also what the benefits and risks of such a high productivity period was. To summarize that, many older drugs are being applied in new ways because new drugs are prohibitively expensive to bring out. As a result, old drugs still under patent are licensed, which increases their cost, especially for new application compared to new drugs. Old drugs that are not under patent are being licensed for their new use, which is expensive but cheaper than old drugs that are still patented with a new patented use. These two facets either make old drugs useful again, but the new application is a costly way, or old drugs with new applications still under that, which creates the drastically compounded price. And that is what makes new drugs so expensive, not the animal experiments that fail. And that leads nicely into funding.
Yeah, we're out of funding. Well, then tell the cancer patients they'll have to wait, Pete.
Very little drug development is funded by public grants. Some is by private grants and bodies, but these are often narrow projects that are working on established research or products. This is seen as a blatant act of emotional blackmail and almost petty. As an example of this is Taxol, a chemotherapy agent. that was identified through public funding through a nationwide program in America. It was then isolated and found to be untenable in that form. They then had to discover a substitute, which they did, but could not produce in mass due to the limitations of the machinery and industry required. Therefore, the license to produce a synthetic form was sold to another company, Bristol Myers Squibb, who now produce it under license, and then it went off patent, which reduced the cost, and as a consequence, you now have a cheaper, widely produced generic product and the original. DNA is now brought up.
I read mice share 97% of DNA with humans.
The 97% of DNA is increased through the use of GMO mice. This leads to mice that have a much closer matching DNA in the area of research with which it should be apparent to this channel, given their earlier reference to the albino mainstay of research mice, the BALB, this being a background mouse used because you can introduce genes to it so easily. This is the breed which gave rise to many subspecies of specialized genes and diseases as a model. And to address the very next point that they make about species differences.
True, but that 3% makes a big difference.
To put it in another context, humans share 99.99% of their DNA with each other. 96% with chimpanzees, 90% with a cat, 84% with dogs, 80% with a cow, 65% with a chicken, and 61% with fruit flies and a banana tree. Considering that chimpanzees are seen as the gold standard of research, 97 is pretty damn good as far as matches go. This is even more true when you consider the genetically modified mice who have had the mouse gene, if it is not the same as a human gene, replaced. This is to be consistent and direct matches with the human variant. A fact recognized by this quote from Van der Warp and company. Failures also result because of critical disparities, usually disease-specific, between the animal model and the clinical trials testing the treatment strategy. And this quote from Mack and company in 2014. In many cases, mouse models serve to replicate specific processes or sets of processes within a disease, but not the whole spectrum of physiological changes that occur in humans in disease setting. That's the end of the quote. This is a most reasonable balance of human safety, animal safety, harm reduction, and reliability and efficiency of the experiment. It is very difficult to completely simulate a disease from humans in an animal, and you are simply unable to experiment on people wantonly anymore. The extreme examples.
In one extreme example from 2006, a new cancer drug was tested in mice and passed with flying colors. But when human subjects were injected with just 1/500th of the mouse dosage, within hours, all of the volunteers went into catastrophic multi-organ failure and had to be rushed to the ICU.
Yes, any extreme example is going to be very bad or very good. And this video would serve itself far better with using a reasonably average example, rather than stating the unpredictable outcome of an immunological response to a particular cancer treatment experiment. More so than anything, given the high failure rates of any clinical trial, extreme examples will not be hard to find. Considering how many clinical trials come about from animal studies, you must wonder about the human and animal models and methods of action. The medicine thing goes back to the genes inserted. You can only have a medicinal clinical trial on the basis of a closely matched animal, and you can only get so close with an animal that's not the same species. One gene can act one way, and if it is a different version, for example a mouse, than if it is a human, this requires a properly designed experiment and understanding of genes and molecular cell biology. Most of the inserted genes are human or the mouse equivalent, which should act the same way. The reason these trials often fail is due to poor integration of research findings or an understanding of the concept being explored. This is recognized in an article by Van de Warp and Company in 2010 to quote, "Translational failure may be explained in part by methodological flaws in animal studies, leading to systemic bias and thereby to inadequate data and incorrect conclusions about efficacy." End quote. But consider that. Even research from 5 to 10 years ago shows that often these failures are not owing to anything within the animal model itself, but to a failure on the part of the researcher to accurately and adequately account for variations within species and their application of the experiment. They seem to gloss over this particular point, but why rodent models?
Because they're cheap and convenient.
The cost of a mouse is between 10 and 50 United States dollars for each. Labs may even breed their own for significantly less. The mouse costs roughly the same amount to keep and feed for a year as it does to buy. They require very little space, are social animals, and are easily handled. The similarity to humans makes them ideal as a substitute for initial investigation. They can also be ordered with special genetic mutations, or, as said, genetically modified organisms. Compare this with the gold standard of a chimpanzee, who cost $60,000 US a year. They require a lot more food, specialized facilities, specialized training, specialized staff, specialized equipment, and more. And we haven't even touched on their cost beyond initial purchase yet. So rodents are not just simply cheaper, they are by far and away practical. Next is their rather misleading and deceptive tangent on animal welfare and ethics.
Finally, the Animal Welfare Act, the only federal law that covers testing on animals, doesn't protect mice at all.
Oh yeah! We can never get away with this on rabbits!
The Animal Welfare Act may not cover mice, but all experiments with animals go through an ethics committee, who are bound by other acts in the legislature. These restrict what they can and cannot approve based on various principles and guidelines, examples of which are the environment, alternative organisms, alternative experiment designs, and tests. These are often referred to as the 3 R's. The 3 R's are replacement, in finding an alternative to an animal, reduction, in reducing the amount of animals you require for the experiment to the bare minimum, and refinement, to ensure that no repetition and no other corrections are required, to mean that no other animals are harmed beyond what is used in that initial number. But they seem to ignore this.
The only federal law that covers testing on animals doesn't protect mice at all.
Animals are covered by other legislation though, and this applies particularly to rodents. They claim that because rodents aren't covered by this, they're not protected or bound by any particular limitations. However, any facility that does receive funding from the public is bound by different laws that are not mentioned in this video. That being the Public Health Service Policy on Animal Humane Care and Use of Laboratory Animals. This requires ethics approval and more for animal-based models and research. Despite the wild claims made in this video, there are significant limitations on what can be done to animals, and not the portrayed moralist, ethicless, violent, criminal type scientists you see here.
Oh yeah! We can never get away with this on rabbits!
As the actions described could quite easily result in criminal and civil charges. Mice are not used because it is easier in all cases, but because it is more accurate. But more will be provided on that at the end. Dr. Azra Raza. The video complains about how expensive drug development is, and then goes into this section about how prohibitively expensive other methods are.
And most other testing methods are prohibitively expensive.
They need to make up their mind. Either the animal models are bad, or expensive testing is bad. They cannot have it both ways. Of course, they do not approve human trials without animal testing first. As said earlier in the video, even tiny samples of promising leads can lead to dangerous results. Imagine if the 1/500th dose from a mouse sample was actually the full amount. It would not end with a rush to the ICU, but death. this section only applies to cancer, then the following is equally true across the board. Alternative experimental designs are the final obvious sign of the propaganda machine behind this video, and it is their expert. Seen here is a small section of their spiel.
Because if we want to cure cancer in humans, we should be studying human cancer cells. We are wasting valuable resources and time on mice.
We do have human cancer cells. Examples of this are the HeLa cell line. This is among others used in cell culture. But cell culture does not have the necessary complexity or interaction of a whole organism. As an example of cell-based studies, take hepatocytes or cardiomyocytes. These two are the primary cells of the liver and heart. Grow them in a flask with appropriate growth media. Over time, divide the population of cells, ensuring that this is done only a limited number of times due to the probability of mutations occurring increasing with each generation. Once you have enough cells, you can then begin an experiment based on an existing body of research. This could be the exposure to chemicals, energy, physical forces, or biological agents. These will cause a change in the cultured cells. As an example, a new chemotherapy lead might be tested. A well-designed study would account for the time, dose, number of doses, and more. These could then be measured against the initial number of cells and the viability, along with other preliminary results. A good researcher then follows this up with what is probably going to be genomic and proteomic research. This will tell them what has changed in the cell, what genes are activated, what genes are deactivated, and more. Fitting these into the existing literature or art, depending upon the funding source, would allow them to understand what they did wrong if the experiment failed, or what they did right if it worked, and further contribute to the body of research, thereby allowing other people to find out where the path works in a more complicated organism. There are also teams working on things known as an organ on a chip, this being a possible substitute for needing to extract only the heart from something like a mouse. Dr. Azra's arguments are addressed in this article by Mack and Company in 2014. Due to practical and ethical concerns associated with human experimentation, animal models have been essential in cancer research. However, the average rate of successful translation from animal models to clinical cancer trials is less than 8%. Animal models are limited in their ability to mimic the extremely complex process of human carcinogenesis, physiology, and progression. Therefore, the safety and efficacy identified in animal studies in general cannot be translated to human trials. Animal models can serve as an important source of in vivo information, but alternative translational approaches have emerged that may eventually replace the link between in vitro studies and clinical applications." End quote. In other words, a mouse is needed to show the complications that are not apparent in cell culture, but in time this will be worked out. This video is all the more aggravating because they make the above claims and they fail to make the above claims, then state that the very reason they are not a problem is the exact redress describing why they are so very useful in the next part of the video.
Eyes. Want a mouse with no immune system? Or how about one that glows in the dark? Extra tumors strike your fancy? You can have whatever model of mouse you want.
Unless this production is advocating for something like anti-vaxxers, through disparaging the translation of medicine and theories of science, proven in animal models and derived from other non-human sources, this video is of little value. All of modern medicine is built on the approach using animals before humans, and there are rules in place as described. These mitigate the larger unrestricted use of animals in research. They require alternatives to be used and prior to animals, and that all safety precautions and minimization strategies are implemented. Through such mechanisms, the best of the best clinical results get through to the next stage of research on humans. This is the best probability of success with the smallest risk profile. The only alternatives are a return to involuntary experimentation on incarcerated persons, such as the Stateville Penitentiary malaria study, low socioeconomic population studies like the Tuskegee syphilis study, or hiring people like Yosef Mengele the human rights abuser. Links to these particular events are in the description box below. The problem is summarized by Leonidas, who was quoted at the beginning. To begin their quote, "Some of the translational failures may be due to difficulties in communication between different fields in the spectrum of basic, preclinical, and applied research. Evidence based on medicine does not seem to have penetrated basic and preclinical science, whilst basic and preclinical research is often performed in a clinical and methodological vacuum." End quote. Or in the words of McGonigal and Riggory in 2014, "The predictive value of an animal model is thus only useful as the context in which it is interpreted. Finally, rather than dismissing animal models as not very useful in the drug discovery process, additional resources like those successfully used in the preclinical PK assessment used for the selection of lead NCEs must be focused on improving existing and developing new animal models." End quote. And this problem was recognized more than a decade ago with this quote from Pound and Company 2004. "Many animal studies were of poor methodological quality," meaning that very little of the failure is owing necessarily to the idea of using an animal, or necessarily to the animal itself, but often to a failure on the part of a researcher to appropriately execute or design an experiment using the strengths and weaknesses of the organism that the experiment is based around, or where they are familiar with the organism in question, they have a shortcoming of knowledge or an inadequacy of the experimental design to be adjusted for the experimental end goals. Thank you for watching this video. Please take your time and watch the original video in the link provided, and please post any comments, questions, or suggestions you may have below.